Hello everyone, this is Dr. Rafael Gutierrez. Welcome to Human Physiology. Today I'm going to deal mainly with the fluid compartments, the idea of homeostasis, some chemistry, and the cell membrane. And I am trying to make it so this Human Physiology lecture series will be usable immediately. So it may not be set up exactly as some textbooks. Now, physiology technically is the study of how the human body functions. And what you have is you have different aspects of physiology. You have the basic cells and how they respond to the environment and how the body itself reacts based on the messages it gets from cells. Now, the whole idea of physiology is you're trying to maintain life, and so you're trying to maintain a regular intracellular and extracellular environment. You're trying to make sure that what's happening inside the cells and outside the cells have a normal range. Now, usually we talk about something called homeostasis. Homeo means same, stasis means not moving. And so in homeostasis, technically, there's a set point and you bounce between a range and you try not to leave with that. Now, if your body ever leaves those set points, a lot of times that ends up causing some sort of pathology. Before we actually begin, we really do need to talk about water because all life revolves around water. And what we need to know, first of all, is water is a polar molecule. By polar, what we're talking about is that it has a positive end and a negative end. It's actually technically a partial positive and a partial negative. Now you'll see me put negatives here and positive. Over here, if you're dealing with water, you're talking about H2O, so your oxygen would be here, your two hydrogens would be over here. And so when we say it's polar, it means it has an end that's going to have a positive end. In this case, it will be where the hydrogens are. And it'll have a negative end where the oxygen is. Now what that means is if we have a lot of water, it will arrange itself so the negatives will be with the positives and it will end up interacting a certain way. In general in general anatomy, in general bio, you talked about how you have adhesion and cohesion, how water can stick to stuff and how it can stick to itself. Uh, there is a video on that in the general bio, so I'll let you do it. Now, what I do want to talk about is for things to be soluble in water, they have to be able to be dissolved in water. And so we have a phrase called hydrophilic. Now, hydro refers to water. So hydrophilic, hydro is water, philic would be love. So hydrophilic would be molecules that love water. If they love water, it means they can dissolve in water. Usually, if they can dissolve in water, they are polar, but there are small molecules that can dissolve in it, whether they're polar or not. Really, one of the things that happens is even with polar molecules, the smaller a molecule is, the more likely it is to dissolve. Larger ones tend to have less likelihood of dissolving, even if they are polar. Now, if we have something that is not dissolvable in water, not soluble in water, we call it hydrophilic. Hydro, I already told you, means water. If phil means love, phobic, a phobia is a fear of, so hydrophobic would be fear of water. So things that are not water soluble are nonpolar, and they would be hydrophobic. Now, there are a lot of other properties of water. Now, one of the big things to remember is water is a universal solvent. And for that, we have to talk a little bit about what a solvent and a solute is. Solvent is technically something that dissolves something. A solute is what gets dissolved. And we'll deal more with that later. The other thing is that it forms hydrogen bonds. If you remember, you have a partial positive side and a partial negative. In the hydrogen sides, you have a partial positive side. And so if you have hydrogens on one side, the the negatives tend to actually be pulled to whatever it's bound to. And so that hydrogen will actually attract or be attracted to things that have a partial negative side. And so that's how you make hydrogen bonds. Hydrogen bonds tend to be weak, but a lot of times there tends to be a lot of them, and that's what gives them the strength is the amount. The other thing that's interesting is it has a high heat capacity, which means that heat capacity is energy required to move temperature. And you actually do require a lot of energy to get water to change temperature. It does tend to spread the heat relatively even, and so as things heat up, water will pick up excess heat. It will warm up. Now what that means is if there's an area that has a lot of water, it tends to not heat as much. The atmosphere tends not to get as hot as areas where you don't have a lot of water. That's why if you're in coastal cities, you tend to have a more cooler temperature during the summer than in inland cities. It also does act the same way with the when it's colder temperature outside, it tends to release heat, but the heat capacity is the ability for it to, the energy required for the water to move 1 degree. Now, the other thing is it also requires energy to change phases. So over here, I actually mentioned the melting and boiling. For water to melt from ice, you need about 334 joules per gram. For it to boil, you need 2,265 joules per gram. And that's actually that's not to get it hotter, it's just so you can actually melt something or boil it. You also have to add that heat if you're trying to get it from liquid to solid. Now, just to give you an idea, 1 calorie is equal to roughly 4.144 joules. So you're actually noticing that this is actually a lot of a lot of energy for changing stuff. Now, that we have water, we can talk a little bit about the basic body fluids. And as we are dealing with humans, we can actually talk about how about 60% of the total body weight is water. So if you have someone who weighs 75 kilograms, and yes, I will be using kilograms because it's actually in medicine, you tend to use kilograms because it's easier to calculate dosage. But if you have a 75 kilogram person, about 45 kilograms of their weight would be water. Now, fluid tends to fluctuate between intracellular, not intercellular, but intracellular and extracellular fluid compartments. And this is based on mainly osmosis, but it does have some aspects of diffusion and some hydrostatic pressures, which we'll talk about later. Now, intracellular fluid comprises 40% of total body weight. So again, if we have a 75 kilogram male, you're talking about 30 kilograms is the water inside the cells. Now, this fluid tends to be relatively well controlled just because of its concentration, but there are different things that can actually happen. Now, the intracellular fluid, like I said, tends to be well controlled. Extracellular makes up about 20% of total body weight, so about 15 kilograms in a 75 kilogram person. And it is made up of the extracellular, the interstitial fluid, which is about 12% or 9 kilograms, and the plasma, which is the fluid found within the blood, makes up about 5% or 3.75 kilograms. Now, I do have actually references here where I got it. It was an article. I couldn't remember the exact percentages, so I always look that up. Now, fluid moves between one compartment to the other. So I want you to think about 66% of total fluid is in intercellular. 26% of total fluid is interstitial, the fluid between the cells and the other stuff. And about 8% is plasma. Now these do change a little bit, but one of the things that can happen is fluid can move from side to side. And one of the things that usually happens is when you're losing or gaining fluid, it has to come into or leave via the plasma. So if you notice, this is the least amount of fluid. That's where you're going to get fluid leaving as you breathe. Yes, when you breathe out a lot of water. If you don't believe me, put your hands together, breathe in it. You'll notice they get wet. You can also lose it from sweat. And one of the interesting things about sweat is you usually reabsorb the sodium, the salts, and so you tend to lose water. You can also lose water through urine from a little bit of GI tract here too. And so you have a lot of different ways. So GI tract, the urine, breathing and sweating. Now the way you get water into here is through the digestive system. You either drink pure water, or you drink water, I should say, not pure, or H2O for water, or you eat, and by breaking down food and using it as a metabolism, you actually convert that into water. And so your water comes in from drinking. And so if you're drinking water, you drink it, it goes to your plasma. From your plasma, part of it goes into an interstitial, and you can see how much difference there is as you go into bigger and bigger places. So you can actually filter it via kidneys, via lungs, sweat, and you have the digestive tract. Now remember, homeostasis, it's moving around a set point. Technically it means same, not moving. It tends to be in the set point and it doesn't move. Now if you think of someone weighing 100 kilogram person and they manage to sweat out 500 milliliters of water, if the body weight is 60% water and a liter of water weighs 1 kilogram, we can actually see how the half a kilogram of water will come out, or half a liter of water will come out of the plasma. And so the amount of fluid you would have here would now be roughly 4.5 liters. Now, once you have that happen, you end up having to get fluid to equilibrate it. The reason is the drop in blood in plasma will cause a drop in blood pressure. And so to avoid having that, you can get the fluid from the interstitium to move to the plasma. So if you think about it, to get it roughly there, you would have to move you would have to actually get 5 liters total, but you're going to get that 5 liters, half of it here, let's say. And so you only need about a quarter of a liter. And that quarter of a liter will also be pulled out of this 40. And so what happens is these would end up helping maintain the plasma levels at roughly 5 liters. Now the other thing that happens is if blood pressure drops, you can cause the blood vessels to constrict and increase blood pressure there. But if we were actually looking at this, let's say we actually sweat 500 milliliters. It initially would come out of the plasma, and what would happen before you actually can get a drink even is the fluid would actually move to these different areas. That's usually based on osmosis, and it's based on the dissolved particles causing a pull. Now before I actually get into dissolved particles, we should talk about basic elementary chemistry. And so one of the things I'm actually going to talk about is an atom, which is at its most basic, most basic, it's just a hydrogen. And what a hydrogen is it's a nucleus which has a positive charge and an electron cloud which has a negative charge. Now, the neutron over here, this big thing here, I'll put an N for neutron for now, and U for neutron, and C for nucleus, I'm sorry. The nucleus is made up of a proton which has a positive charge and a neutron which doesn't have a charge. Now, you can have a variation of neutrons. In this hydrogen, we actually are putting 1, so this would have a molecular weight of 2. Most hydrogens would just have a proton and an electron. Proton has a positive charge, neutron has no charge, and the electron has a negative charge. And the electron, as it's actually on the outer side, it can either be lost, or depending on the atom, sometimes you can gain an electron. As it has a negative charge, if you lose the electron, the negative charge, you end up having a positive charged ion. An ion is a charged molecule, and if you gain an electron, you end up having a negatively charged. So if you remember cathodes being positive, a cation is an ion with a positive charge, meaning it's lost its electron. An anion has a negative charge, which means it gained an electron. If we take water and we were to break hydrogen and water, a lot of times we'd have a hydrogen with a positive charge, giving us a cation, and the oxygen would actually maintain a extra electron, giving a negative charge, and so you end up having two ions. Now, there are certain things called free radicals, and these are highly reactive chemicals where the electron on the outside doesn't match the proton inside. If you actually take oxygen, for instance, has 8 protons. If you have 8 protons and 9 electrons, it's not paired right, and so that free electron will try, that's bound to the oxygen, will try to neutralize itself by binding to other things. That's why free radicals, a lot of times people talk about causing cellular damage. That's how they can do it. They have that extra electron that allows it to react. The way we actually combat it is by using antioxidants. Now, I actually use this picture a lot. It is from the International Union of Pure and Applied Chemists. It's one of the best. I actually love their periodic tables because it's simple. Now, always look at this, and I would tell you to make sure you have a copy of one. And on the hydrogen area, the column with hydrogen, I want you to put +1. Over here where we have beryllium, I want you to put +2. And I want you to skip all the other ones until you get to carbon. And I want you to put plus or minus 4. I'll explain this a little bit. With nitrogen, -3. Over oxygen, -2. And -1 over fluoride. The reason I want you to do that is this actually tells you how many bonds something can actually make based on its outer shell to a degree. And it also tells you that a lot of times these molecules, when they're actually left alone, can end up getting a charge. So when we were talking about hydrogen earlier and how when it's broken, sometimes it loses its electron, and so it has a +1 charge. Magnesium can lose 2 electrons, so it has a +2 charge. Now, when we get a carbon, I put plus or minus, and I want you to think of plus or minus carbon here as something different. Carbon is very important, especially for life. The chemistry of life is usually the organic chemistry, which revolves around carbon. Now, I want you to look at the nitrogen. I'm going to have you skip the phosphate for now. The oxygen and sulfur, and come here. And you can see that these 3 will end up picking up electrons. So the nitrogen usually will have a -3, oxygen and sulfur, -2, and fluoride, chloride, bromide, -1. The other thing I want you to actually, the other ones here, they're called transitional. I'm going to deal with those individually later, mainly because they act weird. For instance, if you look at iron here, iron can have a +2 or a +3 in the human body, and so things can actually change a little. But the reason I want you to also see this is this. If you think of what I told you, carbon being the molecule that revolves around life, you can see it's plus or minus. And this plus or minus can tell you that it can bind to either positives or negatives. Well, negatives can also bind to negatives, share the electrons. But what I mean by that is this. You actually have 4 areas where electrons can come in here. And so, or 4 areas where you can share electrons. So if you have carbon and 4 hydrogens, that would actually fill the shells of carbon and hydrogen, so they'd be neutral. If you go to carbon and you put it with oxygen, you can see that a carbon molecule will actually bind to potentially 2 oxygen, giving you carbon dioxide, and it would be neutral. If we look at oxygen being a -2, it can bind to 2 hydrogen, giving you water, H2O. And that's actually one of the really neat things that can happen. The other thing is, sometimes we will see some of these things free, And this tells you what charge they would have. So if you see a sodium ion here, it usually will have a positive charge when it's in water. Potassium will have positive 1. Chloride will have a negative 1. And you'll see that more and more as we go along. Now I love this Brondo thirst mutilator because I'm a big fan of the movie Idiocracy. The reason is because a lot of times people sell you stuff without, and pretty much people buy it because, well, the best way to put it is take your average person. If you don't know what I'm talking about, Google George Carlin and see what he says about the level of people's intelligence. Now, what you have is you have salts and you have electrolytes. Now, salts, what they are is they're compounds made of a cation and an anion. Usually it ends up happening because a salt and a base and an acid came together, but not always. But what happens is these are compounds with positive and negative ends. And a lot of these, when they're put in water, they come apart forming ions. So if you think of sodium chloride, sodium tends to have a net plus charge, chloride has a negative charge. When you put them together, they're neutral. But if they go into water, they disassociate, and you have positive sodium and a negative chloride. Now, electrolytes are substances that come apart in water to form ions. So just like salt, right? Well, it doesn't just have to be salts, it can be acids and bases as well. Now, about blood. Blood is a solution, and I told you I was going to talk about this in a little bit. What a solution is it's a solvent with a solute. In the case of blood, the solvent would be water, the universal solvent. The solute would be everything else that's dissolved in there. Now, the other thing we want to talk about when we're dealing with blood is the osmolarity. And osmolarity is how many osmoles there are per liter. Now, this is what usually happens. If you take a mole of sodium chloride, it means you have Avogadro's number of sodium chloride. But when you put it in water, it doesn't react as sodium chloride. In water, it's going to act as sodium plus chloride. They're going to split up. So if you had a mole of sodium chloride in a liter of water, you would have 2 osmoles. They would act as 2 molecules instead of 1. So we actually want to look at osmoles, which is the number of moles per solution in liters. The osmolarity of blood tends to be about 285 milliosmoles per liter. That is actually important because if you have lower osmolarity, it means you're going to have less dissolved particles, and so water is going to act a certain way. Another thing that's important to know is pH, which stands for potential hydrogen. P for potential, H for hydrogen. And what I'm going to do is I'm going to make this H a capital. So you can see pH, potential hydrogen. What pH is it's a measure of hydrogen, and it's actually in a base 10 log scale, meaning that the more hydrogen ions you have, the lower the pH. And this is the way it works. Neutral pH, as found in perfect water, would have a pH of 7. So pH of 7 is neutral. An acid would have a pH of less than 7. So if we have more hydrogen ions, you'd have an acid and you'd have a pH of less than 7. A base means you have more hydroxide ions. If you think of water as H2O, hydrogen ions would be H+, hydroxides would be 1 oxygen with 1 hydrogen, and so you'd have a negative. And this, if you have a higher amount of hydroxide ions, it means you have a higher pH. It can neutralize acids pretty much. Now, when we deal with blood, we actually will notice that the normal pH of blood is about 7.4, 7.35, 7.45 pH. That's what we call normal physiological pH. Now, besides blood, in blood you do have a couple different things. One of it's the plasma. And plasma is the fluid. And so plasma is a fluid, and it's made up of salts, minerals, any dissolved inorganics, water, and also circulatory proteins. Now, I will tell you, if you get rid of the circulatory proteins, so if we were to take blood and we spun it down, we can end up getting the plasma. We can actually then also remove the circulatory proteins, get rid of these. And what we are left with is the salts, minerals, and inorganics with water, and that would give us serum. So serum is pretty much the fluid part without the circulatory proteins or cells of blood. Now, like I said, the plasma is divided into two parts. You have the plasma proteins and the serum. And over here you see the normal concentration of ions found in blood. So you have sodium, potassium, chloride, calcium, bicarbonate. And so these actually have a certain concentration which is considered normal. If they are not in their normal range, if you notice I have plus or minus here, then you actually end up getting pathologies, but we'll deal with those later. Today, mainly we're going to deal with sodium, which is here, Na, and potassium here. Okay, and we may use some bicarbonate too. Now, besides that, when we are dealing with chemistry, we should actually talk about some of the different bonds. And I'm going to start down here with hydrogen bonds because I've mentioned them before. They tend to be the weakest of the chemical bonds, and they are usually between a partial positive and a partial negative, like you find in water. Ionic bonds are bonds due to electrostatic charges, which are broken. A lot of these are broken in water. If you think of salt, sodium chloride, they're strong bonds, but when they get in water, they disassociate, and when you remove the water, they come back together. Covalent bonds are the strong bonds. They require energy to form and release energy whenever you break these bonds. So whenever you're dealing with energy, you're usually dealing with covalent bonds. Now, as we're dealing with putting things together and breaking it down, we should just mention metabolism. And metabolism is a combination of building things up and breaking them down. When you break stuff down, it's called catabolism. And when you break things down, you end up releasing energy. And we call the release of energy, as it's in heat, exothermic reaction. Exo, you can see here, exo, I put it alone, means outside. Thermic means temperature. So you end up getting temperature released outside. Now I will tell you, to break the bonds, you do need activation energy, which starts getting the bond excited so it could be broken. But at the end, the total energy that gets out is energy coming out of that. Anabolism is when you build stuff up. And to build a bond, you need to add energy, not just an activation energy, but you need to add total energy to form the bonds. As you're putting energy in, we call that a endothermic reaction. Heat or energy is going in. I would tell you to remember endo and exo because you'll see it again. Now there are different types of reactions. One of the reactions that a lot of times we talk about is a reduction reaction, and then or hydrolysis, and then we have the oxidation. In hydrolysis, what we are is we're breaking using hydrogen. And so if we take hydrogen and we have a CHO3 here, we can end up taking that hydrogen, we usually have about two of these, sorry, and you would break the bond, which would give you some energy, and you'd end up making carbon dioxide and water. And that carbon dioxide and water will pretty much go on its own. It's paired even, and it will actually give you some energy. Oxidation, you have oxygen, free radical, you have iron, and what you have is oxygen binding to something to neutralize the charges as well. Besides that, we do have also things called dehydration reactions. Dehydration reactions means you're removing water to link things here. Now, I'm actually going to use a linking of sugar. And so you can see a 6-carbon sugar here, another 6-carbon sugar here. Over here, you're noticing that water is coming out, so we're losing water. And over here, what we have is we have a bond forming between these two sugar molecules. And so that's a dehydration reaction. Can use it for a lot of things, not just for sugars. Now, I also want you to pay attention to something called hydrocarbons. Now, if you look at this, hydro for hydrogen, and obviously carbons for carbon, these tend to be non-polar molecules, meaning that they do not dissolve in water. They tend to be relatively neutral. They don't have a partial positive, partial negative. It's going to be a molecule that's mainly neutral. And a true hydrocarbon is composed of only hydrogen and carbon. Again, they're not soluble in water, so a lot of times we call them, oh, wrong one, lipophilic. I grabbed the wrong thing. Lipophilic. So lipo means fat, philic means loving, so hydrocarbons tend to be lipophilic. And one of the neat things about carbon is they can form different types of bonds. Remember how I told you carbons are the molecule that we tend to deal with mainly when we're dealing with life, with this and water. And so anytime you hear organic in chemistry and biology, you're usually referring to something having carbon. And it can share electrons, and it can share 4 electrons. So if you're using hydrogen, for instance, you'd be able to put 4 hydrogens onto this one carbon, or you could do 3 hydrogens and have another carbon next to it. Hydrogens will only find these 1 bonds, but there is actually something else that will happen, which I'll tell you a little bit. Now, one thing I was going to tell you is you can actually, in some of the carbons, you can actually have double bonds, which I'll show you later. Now, besides hydrocarbons, you also have carbohydrates. And if you think of hydration, it means water. So a carbohydrate is a molecule composed of carbons and waters. So carbon, oxygen, and hydrogen. These are usually referred to as saccharides, and saccharides are sugar. If you have a monosaccharide, mono means 1 sugar molecule, and you have 2 different types of monosaccharides. You have hexoses. Hex means 6. Os is sugar. So anytime you see OSE, think of sugar for now. And a ribose is a 5-carbon sugar. So when you're actually talking about ribose nucleic acid, ribose tells you it has a 5-carbon sugar and it has a nucleotide. Now you do have polysaccharides, disaccharides, and what they are is a disaccharide would be 2 monosaccharides which have been linked together through a dehydration, polysaccharide, many of these. Now, I mentioned hydrocarbons, but I want to get a little more specific to a special one called fatty acids. Fatty acids are hydrocarbons with a functional group called a carboxylic acid. Usually the carboxylic acid will have a carbon. You'll have a double bond with an oxygen. And you'll have another bond, which is a oxygen and a hydroxide called a hydroxyl group. And this would be bound to something else. We'll put R for functional group R. And so this is an acid, which means that hydrogen can actually be taken off. And so when we're talking about fatty acids, we're talking about hydrocarbons with this carboxylic acid group. And we do have 2 different types. We have saturated fatty acids and unsaturated. Now, a lot of times people tell you, oh, unsaturated are better, and they are. They actually do find that unsaturated tend to have more benefits than saturated. But what we're actually talking about is in a saturated, what it's talking about is all the carbons in the fatty acid chain have the maximum amount of hydrogens you can expect to have on them. So if you have, let's say, 2 carbons, all of them would have hydrogens, single bonds with hydrogens. There's actually something I told you about double bonds. So you have hydrogens, hydrogen here. And so that, as this goes on, this is all you would have is carbons with as many hydrogens as you can fit in here. I'll put a H here if you want. Put a H. There we go. Make a short chain fatty acid. O H. Okay. And so this would be a small saturated fatty acid. Now, a lot of times what we can do is we can form double bonds. And if we form a double bond here, what we end up with is an area where you should potentially have a space for another hydrogen. And so this is what we call an unsaturated. Now, one thing you'll notice is the two hydrogens are on the same side. And so we call this a cis fatty acid. If, on the other hand, we had a carbon double bonded with a carbon over here, and the hydrogen, one of them was up here, the other one down here, this would no longer be cis. This one goes across. So this would be a trans fat. Now, trans fats tend to be synthetic and are used as food additives because we like the taste of them, but they are linked to many diseases. And a lot of countries around the world have actually limited the amount, by law, the amount of trans fats that can be added to anything. Not us here, don't worry about it. The US, you can kill yourself with fatty acids and triglycerides and trans fats. Now, there are other lipids to look at. One of them is called a triglyceride. A triglyceride is a glycerol molecule. And a glycerol, it's a 3-carbon structure. And a triglyceride is a glycerol with 3 fatty acids. So when people talk about triglycerides, it means you have a 3-carbon with fatty acids hanging off of them. A cholesterol is a lipid in a ring configuration. So instead of being straight fatty acid, it's actually usually in these steroid rings. And they are used to, they actually do have an alcohol group, and they are used to stabilize the cell, what's called the cell membrane. The other lipid that I want you to pay attention to right now is one called a phospholipid. Lipid tells you it's a fat. A phospholipid tells you that it has a phosphate, and a phosphate is a phosphorus with 4 oxygen on it. And these are linked to fatty acids. The interesting thing about that is the phosphates are water soluble. The lipid tails, the 2 lipid tails that it's attached to, are fat soluble. And so what you have is you have a fat soluble head, the phosphate, PO4, and you have 2 fatty acid tails hanging off of them. And as this has fatty acids and phosphates, it's referred to as an amphipathic molecule, meaning that it can dissolve in both water and fat, or it's afraid of water and afraid of fat too. And so what happens when you have a lot of these and you put them in water, they're going to arrange where you'd have the phosphates on the outside. Then if you would end up making a sphere with phosphates on the inside. And between the phosphates, you would have your fatty acid tails here. Fa for fatty acid. And so you have what we call a phospholipid bilayer. What that means is if this were in water, water would have a hard time trying to get into this. Any water-soluble molecules would actually not be able to get in. And if we started this with the same things inside the water as outside, that'd be fine. Now, that we talked about the phospholipid bilayer, we do have to talk about proteins. And proteins are made up of amino acids. And amino acids are aminos, which are a nitrogen group. You can see it here, a nitrogen with 2 hydrogens. and an acid. And you'll notice that the acid that we've been dealing with and we'll deal with a lot is this carboxylic acid group. So what we have is we have a carbon with the oxygen here, hydroxide here, and then we usually would have some other carbon. And then on that carbon, we would have our ammonia group. And so this would be the most basic amino acids, which is called glycine, which we'll talk about later. But whenever you have multiple amino acids, you can take the hydrogen off one and the carboxyl off the other, take them off, form water, and bind them together. Now this would not bind to this one. You would have another nitrogen with hydrogen here. So this hydrogen and this hydroxide would come together, leave, and what you end up with is you end up with a bond called a peptide bond. Between the carboxylic acid and amine group form this dipeptide, and many peptides formed together will end up forming a protein. Now, amino acids usually are hydrophilic, but you do have some amino acids that have hydrophobic side chains. What that means is if there's a protein that has a fat loving place, it's going to try to be away from water. So, and it's going to change the way it actually folds. And there are different types of folding you have in protein. But before I talk about that, I want to mention the two structures, primary structures and secondary structures of proteins. You have the primary structure, which is the sequence of amino acid, meaning you have an amino acid, it's bound to another amino acid, it's bound to another amino acid, and so you have the primary structure, which is just what amino acid is in position 1 and 2 and 3. The secondary, you can either have beta sheets, which are, these proteins may actually form these pleated sheets like that, or they can end up forming spirals or helixes called alpha helices. And so these are your secondary structures. Now the secondary structures can come together, so you may have a sheet, then a couple pleats, then you'll have another helix, and so you can end up having a tertiary structure, and then you can actually even modify it where you can cut parts of the protein, and that part that you cut may have actually only been used as structure. And so you have secondary, tertiary, quaternary structures of proteins. Now, proteins have a lot of functions. You do have structural proteins, which just give structures. You do have enzymes, which an enzyme, what it is, it's a protein catalyst. And a catalyst is something that speeds up a reaction without being used up. The way it does it is by decreasing what's called the activation energy. What it is for you to actually do something, for you to either make something or break it, you need to put some energy so you can do something with it. And that energy put in before you get out the energy or put in the energy you're going to need to is called activation energy. There's more on proteins I'll talk about next video. So we do now have a phospholipid bilayer. And one of the things I want you to know is the cells are the basic unit of life. And in humans, the outer layer of the cell is made up of a phospholipid bilayer with many other structures put in there, which we call a fluid mosaic model. It's fluid because it is moving, because of the way the phospholipids, which is the main component, are. It's mosaic because it has proteins. Some of them go all the way through the membrane, which I'll talk about next time. They're called transmembrane if you want. There's cholesterols, which help stabilize the phospholipid bilayer. There's glycoproteins, proteins with sugars on it, lipoproteins, fats with sugars on it. And you have a bunch of different things in there. And so we call the cell membrane the fluid mosaic model. Now, one thing about it, it is selectively permeable, meaning it'll allow some stuff to go through and not allow other stuff. Now, when we talk about this, we really need to talk about two things. One is osmosis, and the other is diffusion. And we have different types of diffusion, which I'll talk about in the next video. But osmosis is pretty much water moving through a semi-permeable membrane due to the differences in osmolarity, meaning what's dissolved inside is different on one side of the cell than the other. And if that happens, then fluid, water, will move across the membrane based on certain transmembrane proteins. Diffusion is movement of particles from high to low concentration based on what's called Browning forces, which you learned in general bio. Now that we have all this, we can put this together. And let's start with the blood plasma to begin with. Now this will not stay the same in red blood cells, and you will actually change this because by urinating or getting more or losing it, or actually binding it to stuff. So what we have is we have blood. We're going to start with blood plasma, and let's assume that the entire white area here is serum. It's exactly like this, and we're going to move different things. But first, we want to take a bunch of phospholipids and throw them in here so they make the phospholipid bilayer. We would have some proteins in here. I'll make them red, that go all the way through, which are transmembrane proteins. We have some peripheral proteins that are just here, integrated protein. We have some proteins that do other stuff. Now, if the serum here, if the serum and the cell here have the same concentration, we can talk about their tonicity. And the tonicities can either be, if they're the same, it would be a isotonic. Iso means same. And that means that fluid will actually move both in and out, but it will be at the same levels. So you won't have a net gain or loss. It's just fluid's gonna move in and out. That's it. And that's what happens in an isotonic solution. If the fluid outside is has more dissolved molecules than the area inside, then the fluid outside would be hypertonic. If it's hypertonic, that would mean that as you have more molecules outside than inside, that would mean that you would actually pull water out of this. That would be in a hypertonic, where you have more solutions outside than inside. Hypotonic, hypo means below, so you'd have less outside than inside, so water will move inside. And so that's actually when we're dealing with the osmolarity of a liquid, we can see what happens if it's isotonic, hypertonic, or hypotonic. And I am actually giving you this area that tells you how much of what you have. Inside the cell and outside the cell, it will be different, but I don't want to get into that until later. Okay, so tonicity and how this affects osmosis. Again, I've mentioned if a cell is put in an isotonic solution, that means that everything stays the same. If a cell is in a hypertonic solution, that means you have more molecules inside the solution that you put the cell in than outside, and so it's going to pull the fluid out or cause it to crenate, means to shrink. If a cell is placed in a hypotonic solution, that means the fluid will go in, causing it to swell and potentially burst. I'll talk more about that as we get into this. Now, what I wanted to show you is how you can use this in a normal clinical setting or a athletic setting. Now, during strenuous activity lasting for more than an hour, athletes can actually lose 2% of their total body weight without any significant issues. This is usually due to water loss. Now, if an athlete weighs 100 kilograms and they lose 2 kilograms or 2 liters of water without any clinical issues, that means that must have been some sort of compensation. You think about it. You have about 5 liters of water. If you lose 2 liters, it should end up causing a decrease in pressure. And so first thing you want to actually ask yourself is, which of these 3 fluid compartments did the water loss come from? And if you remember, if you're sweating, it comes from the plasma. If you're breathing, it's coming from the plasma. So if someone lost 2% fluid, it probably came from the plasma. Now, if you lose 2 liters here, you can end up going to these different things. So if it's a 100 kilogram person, you're talking about 60%, so 66% of the fluid is here. And so this fluid will actually, from the plasma, will actually end up being compensated a little bit by the fluid in the interstitium, and the interstitium will pull the fluid out here. And so what you're going to have is you're going to have diffusion, osmosis, movement of water through semi-permeable membranes across this. Now, the cells inside and any cells that are meant to see changes will actually see a change in osmolarity. You lose water, the osmolarity here goes up. Remember, you lose water, the osmolarity, the osmoles stay the same, so you have the same amount of particles and less water. And so if the fluid is hypotonic, what would happen to the rest of the fluid is it would actually shift within it. And you notice that the amounts, so you're talking about less than 50% is in an interstitial compared to the intercellular. Less than about a third is in the plasma. And so you end up having these, the ability for the body to compensate without losing a lot of function. Now, based on osmosis and diffusion, what would happen to the neighboring fluid? You could see it here. You may have some diffusion occur also, by the way, from the plasma to the interstitium, and the interstitium can come in here because of different channels, but don't even worry about that right now. We'll talk about those later. But you could potentially have that, but what would happen is it wouldn't really affect the intercellular fluid as much as you would think. Now, if the fluid inside, what would the fluid inside of a cell, how would it respond? It would actually move out, right? If the cell above, the cell we were talking here, where it's shrinking, monitors osmolality, what would this athlete feel? Well, if your blood is getting saltier, you would actually see that, and what you would want to do is increase the fluid. And the way you increase the fluid, the only way you can actually increase fluid is through the digestive tract. So you'd want to drink more water, so you'd feel more thirsty. And what would be the best solution to return the fluid osmolality to normal? Well, if it's a hypotonic solution that you're losing, it means that you're losing more water than salt. And so what you want to really replenish in this case would be water. Remember, this is actually looking at losing 2 liters of water in an hour. If you're losing 2 liters of water in an hour, that's a lot of sweat. Okay, so the second clinical vignette I want you to actually think about is something that we all do. Eat something really salty and then go to sleep. Now, if you think about it, the salts would come in, again, through the digestive tract to the plasma. You'd increase the salt, and that would actually cause fluid to be pulled over here through osmosis, and these new salts to also move to the interstitium through diffusion. It would actually not, again, you notice that it's not going to be that big of a deal, and you'd have the same thing happen. Osmosis, water would move over, diffusion, you would have sodium move in a little bit. And so what you would see is, you would see not a change necessarily of volume, but of osmo. And so as fluid is moving over one way or another, what would happen is the intercellular volume would actually decrease because you're pulling fluid out. And what that would do if the cells that were being dehydrated because of the salty food, what they would do is they would sense too much osmolarity, and so they would actually try to get you to drink more, and that would also end up leading to increase in plasma, and so to more pressure, you would actually want to get rid of the fluid, so you pee more. Now, so the sensation would be it actually drive thirst. Now, if you don't believe me that this would happen, do something that I love to do. Have pizza tonight, and pepperoni in particular, and see how often you have to have water before you go to sleep. Okay, here's another vignette I want you to actually think about. A person actually decides to drink 20 liters of water in a sitting. This is horrible. Think about it. You're talking about 20 kilograms of water. So where does this go first? It goes into the plasma. That fluid is going to rush into the interstitium, and it's going to pull the electrolytes from the interstitium to the plasma. It's also going to do the same thing here to the intercellular fluid. And in the intercellular fluid, your electrolytes, your solutes will actually go into the interstitium. And so what's going to happen is you're going to end up having swelling of the cells or the interstitium. And if you have something that swells and it's in, let's say, the skull, they won't actually do so well. So this actually tells you how would this affect the cell. Well, it can cause them to swell and cause them to lose the solutes and not function properly. Now that we have that, let's talk a little bit about an enzyme that can break apart something called a adenosine triphosphate. Now, we usually refer to it as ATP, and for some reason, some people tell you ATP is energy. It is not energy. Energy is actually in the bonds, the covalent bonds between the phosphates. ATP stands for adenosine triphosphate, and what it is it's adenine, which is a nucleotide, and a ribose. A ribose is a 5-carbon sugar, remember? And you have 3 phosphates attached to it. And within these 3 phosphates is where you have the high energy bonds. Now, if you have ATP here, and you break the bond, you end up with ADP and a free phosphate that's not bound to anything. And you get energy. That energy can be used to do work. So ATP goes to ADP. In this case, the D stands for di, which is 2. So adenosine diphosphate would tell you have 2 phosphates for every adenosine. Now, I want to talk about a special transmembrane protein called a sodium potassium ATPase. Now, trans means across. So transmembrane protein means a protein that goes all the way across the membrane. This one is called sodium potassium ATPase. So it's going to deal with sodium, which I wrote here as an A, is sodium. Potassium is K, ATP, adenosine triphosphate. ACE is an enzyme that breaks it. And what ends up happening is sodium potassium ATPase breaks 1 ATP and uses that energy to pump 3 sodium out and 2 potassium in. What that means is if we started with the same concentration of sodium inside the cell as outside, we're now pumping 3 sodium out and 2 potassium in. If we're pushing 3 out, of sodium, Na, and we're pumping 2 potassium in, what it's telling us is the concentration of sodium will become, this usually we'll see concentration of sodium outside the cell will get a lot larger than inside. So this concentration will be higher. The things that I actually showed you, the sodium concentration outside is the plasma. So that's actually the sodium that the cells have pumped out, and the potassium has been pumped in. Now, using the periodic table, which I'm going to show you later, determine what the charge of sodium and potassium are. You ready? So all you have to do is come to this table. If you remember, this is +1. This one is +2. I'll put 1, 2 there. So you now have +2, plus or minus 3, plus or minus 4. I'm sorry here. I'm just going to put 4, minus 3, 2 and minus 1. And we can see is sodium is here, potassium is here. Both of them have a positive 1 charge. So as we move on, you have a positive charge, plus 1, let's put a plus here, and a positive here. Now, as we're pumping 3 sodium out and 2 potassium in, we can see that we're pumping 3 positives out for every 2 positives that go in. And so if we're getting 3 out for 2 in, what we're having is we're having a net loss of positive charges. So based on the charges, the their negative charge would be inside the cell. That means the cell is polar. It has a positive end and a negative end. What would have a higher concentration of sodium, inside or outside? Well, if we've started pumping sodium out of a cell constantly, the sodium concentration outside the cell will increase while the sodium concentration inside the cell will decrease. So sodium now has a higher level inside than outside. Potassium would have the opposite. The inside would be higher than outside. The high concentration of potassium would be higher inside than outside. Based on diffusion, what that means is, well, if you have more sodium outside than inside, and diffusion is a movement of molecules from higher concentration to lower concentration, that means sodium will want to try to go into the cell. If it got kicked out, Na wants in. Potassium, on the other side, wants to leave. Now, let's say we have another transmembrane protein, and we'll call this transmembrane protein a sodium channel. It allows diffusion of sodium through the channel, something called facilitative diffusion, which we'll talk about later, when the channel's open. If we've been pumping sodium out, potassium in, and a sodium channel opens, what would happen with sodium as soon as the channel opens? Well, it would flood into the cell. Now, if we have a negative charge because we've been pumping sodiums out, 3 sodium out, 2 potassium in, and we allow sodium to rush in, what would happen is you would lose that negative charge. So let's talk about what's happening so far. ATP is constantly pumping sodium out and potassium in. And what that does is it gives you a charge across the membrane. And I'm going to put the number minus 70 millivolts. And for this particular cell, when it's at rest, when it's not doing anything, when it's just sitting there, it's pumping sodium out and potassium in. And that charge difference gives you a charge difference of minus 70. A negative charge, and we call that a resting membrane potential. It's a charge on the membrane which makes the cell membrane polar. It has a positive and a negative side. If we open a sodium channel made of proteins, which are transmembrane, it's going to allow sodium in. If sodium comes rushing in, it's going to get this to reach roughly 0. And so it's polar here. When it comes up here, it's depolarizing. It's losing its polarity. And when it gets to 0, the sodium channels close and potassium channels open. And potassium you have higher concentration inside than outside, so potassium will want to leave the cell, causing the charge to come back. And so that's your repolarization here. I'll put R for repolarization, D for depolarization. And then you have once you actually get the repolarization, you then close your channels, you continue using your sodium potassium ATPases, and you reset your cell to have it polar with the right concentrations. Now, as we have a depolarization and a repolarization, we call this an action potential. Now, that is actually the end of this first lecture. What I am going to do is I'm going to show you a list of terms that we talked about, which I recommend making this list on a paper, so when you watch the next videos, you have these ready and available, so you can just jump to any terms that I've mentioned without having to wonder, wait, what did that mean? I hope you enjoyed this and have a nice day.